I have roughly half of the team members are research staff, half of the team members are PhD students. So I do receive research grant from the federal government. I also have a company that I co-founded. So some of the data I actually acquire from my startup company. So the overall research theme focus on two technologies. Visible OCT, so I'll explain why visible. And OCT, I'm sure everyone here knows or heard of that. The other half of my life is focusing on super resolution microscopy. It's called single molecule localization microscopy. I'll briefly touch on that, and I'll explain what this is and why we like it and how we combine super resolution with OCT. The scale, when we say multi-scale, the scale is from 1 to 5 centimeter, which is human eye, to 1 to 10 nanometer, which is single molecules. So that's 10 to the power of 7. And if we zoom in, so this is human eye that acquired by our machine, visible OCT. It's not my eye. My eye is not as healthy. And this is a chromosome from a mouse-derived cell. And that's the 10 to the power of 7. Also, when I explained to my colleagues in the US, this is the signature building. If you have been visiting Chicago, this used to be the tallest building in the world. I mean, years ago, it's called Sears Tower. Now it's called Willis Tower. If we zoom in, same ratio, 10 to the power of 7, you see a single blood cell. So that's the whole range that we're talking about. Now come back to the basic anatomy. I want to show this picture trying to emphasize the difference between human eye and mouse eye. And this actually imposes lots of design challenges for optical systems, especially using OCT. For mouse eye, it's essentially a big glass ball. And it occupies more than half of the posterior segment or the whole globe. So in other words, this glass ball introduce a big aberration. It's very hard to compare with the optical quality of a human eye. So human eye optical quality is much better, although you hear aberration is still bad. But mouse eye is horrible. And we have to design very different optical system to accommodate the poor optical quality of the mouse eye. So a very brief history of OCT. 1991, a young scientist, David Huang, he was an MD PhD student at Harvard/MIT, and he worked with Carmen Profito and James Fujimoto. So that was his PhD thesis. At the time, that was the quality. I'm sure nobody's gonna like it, but it's published in Science, and it's the most cited paper in OCT. At the time, the wavelength was 830 nanometer. The 830 nanometer wasn't selected scientifically. It was whatever available on the market that they can buy. So it has certain historical reason why 830, 840 is the dominant wavelength for the current clinical machine. It's because of optical communication. That's why. So this is the latest one. Center wavelengths around 1040 to 1330 swept source OCT. So over the years, that's a technology migration, and it greatly improved the quality of the imaging. But I'll focus on the two wavelengths, right? 830, 840, 1040 to 1330. Now, we move to a shorter wavelengths. Now, if you focus on here, we're talking about 510 to 610. So this is almost half of the wavelengths compared to the longer wavelengths. And in 2013, we published the first paper. And then 2020, there are a lot more groups around the world start catching up. And these wavelengths introduce two major benefits. That's high resolution and high contrast. So I'll explain what do I mean by high resolution and high contrast. So this is the first paper published in 2013, as I mentioned. At the time, there were not even fiber optics component for this spectral range because nobody's using that. We cannot even buy fiber optics, the connector, the fiber coupler, anything, just doesn't exist. So we have to build a free space OCT. Everything is glass based on the optical table and image it. GE was the lead author on that paper. He just received tenure this year at Hopkins. So this is the bandwidth from 510 to 610 nanometer at 3 dB bandwidth. It transformed to a point spread function, which is a resolution in air around 2.9, around 2-ish nanometer in air. And if we scale by the reflective index of biological tissue, which is 1.33, we have around 2 micron spatial resolution in the eye. And I'll give you a rule of thumb. The OCT, the most clinical machines are from 5 to 10 axial resolution. That's the ballpark estimate. So if you look at, these are from the mouse. This signal looks like random signal. So this is a spectrometer signal. What does OCT do? OCT sends a beam of light to the eye, and the different layers of the cells reflect the light back to the detector. Because of the light are reflected at different depths, a OCT build a interference device to detect the passage difference and to reconstruct the image. So this is how the interference look like. And from the interference signal, we can do a Fourier transform. Of course, there are lots of intermediate steps between that. We can do a Fourier transform to acquire a 1D signal that's called A line. And A line reflects the location of different reflection already. And we align different A lines to form a B scan, and then align different B scans to form a whole 3D. So that's what you see from the presentation. And I want you to look at these small bright signals. So these are actually capillary without doing any angiogram at the time. In 2013, angiogram wasn't even commercially available. We can visualize that. So that's the high contrast benefit. And this is the latest iteration of visual CT and visual CT angiogram. I will show a few cases of how we did it. What you see here, so the green colors are actually ganglion fiber bundles in mouse eyes, and the red colors are capillary network. They're all scanned by the same protocol from the same volume, just process them differently. And because of the high contrast and high resolution, we're able to visualize that. Near infrared is not that easy, I would say, and especially to quantify the single fiber bundles is not that easy. By registering multiple B scans, the OCTA angiogram, the OCT angiogram using visual CT is also quite good, especially getting rid of all the defocusing by doing a better registration. Is referred to as TSA, temporal speckle averaging. What you see here, so these are temporal speckle average B scans from a mouse eye. I mean, I can say this is probably the best quality of the mouse eye any OCT can acquire. And what you see here, so I'll just give you a few results, and I'll explain what these things are. And these are outflow pathway. So the outflow pathway controls the liquid inside the eye being generated and being pumped out. If the outflow pathway has any increased resistance throughout the whole pumping process, the fluid cannot be pumped out. Fluid will accumulate inside the eyeball. When the excessive fluid accumulation, the pressure goes up. And when the pressure goes up for a long time, that's glaucoma. So if there is a way to measure the flow, we'll have a much, much better way to visualize the pressure pass directly. So Si Yu contributes significantly to the small animal imaging. He's already started his tenure track at Oregon Health Science Institution. So how are we doing the imaging around glaucoma? So I'm using glaucoma as an example. This is the signal pathway for optical signal being converted to neuron, and then from the neuron being synthesized by the bipolar ganglion and then passed to the brain. We focus on the synaptic layers, we focus on the ganglion layers, we focus on the fiber bundles, and we focus on the oxygen consumption. So I will show case by case. This is the fibrogram. So we call that fibrogram. In mouse and in tissues, we can resolve individual fibers. So what you see here are a movie, 2D movie of unfast movie of consecutively acquired OCT volume. And then from here, we can quantify the fibers and vessels altogether by doing texture analysis. But this is a lot more straightforward because fibers are sparse in rodent eyes. In this case, the fiber bundles and the vessels are mixed. There's no way to separate them. And now we have to do a OCTA protocol. Now the fibers are gone, right? So they're all from the same data set. One processing both vessels and fibers, a second processing only vessels, and then combine them. There is a very good visualization of bundles and vessels. Now that's the foundation for quite a few of our analysis in rodent animal. We are doing that in human. We haven't published that yet because there are lots of challenges. So for the small animal OCT, it's hard to achieve a large field of view because of the glass ball. So the walk around is acquire 1, 2, 3, 4 scans by tilting the beam and then combine them. So here are combined scan. And once we combine, how we analyze the data. So using the optical disc as the center of the circular and draw circles with different radius one by one. So these are circles with different radius. And assuming these are fiber bundles as a star shape, the fiber bundles are thicker at the close to the optic disc and become smaller going to the periphery area because bundle combine to each other and then goes to the optic disc. And we can count how many intersections between the bundle and the trajectory and draw a curve. So this is the radius. The vertical axis is the log value of the number of intersects. And these two are simulated with high density and low density. So they show up differently at the decay rate. So this is actually a quantitative measure of the fiber bundle density with respect to the radius. And this can be done at a global area, or in most of the analysis, people quantify the eyeball into 4 quadrants, right? Nasal, temporal, inferior, and superior. Now we can do that as well. So without going into the details, all these are recently published, and then you're very welcome to take a look. It's a lengthy analysis, lots of mathematics behind it. But please, if you're interested, take a look at the paper and then contact me if you have any interest or question. So as I mentioned, rodent is easier to access, it's easier to manipulate, but it's too far away from primate because rodent doesn't even have a fovea. There's no vision center. So it's getting more popular in the research community using tissue. Tissue may not be available in the Hong Kong area, but tissue is like a squirrel. It's bigger, much bigger than a mouse. Has an average body weight of several hundred grams. A mouse is 20 to 40 grams. Tissue is like a big rat, but it's closer to primate than rodent. So in other words, it has a quasi vision center. Although it doesn't have a fovea where it's gonna bend and has an avascular zone, but it has a quasi vision center. It's mimicking human beings. Also, the fiber densities are much higher. It's also similar to primate. So it's a good model for us to test whether the individual fiber bundle analysis, especially the K curve, can be done potentially in humans and using tissue as a model. So this is the OCTA fundus. And also, if you look at the quality here, this is the mouse fundus. This is a tissue fundus. It's closer to a primate. The eye quality is much better. So this is the OCTA quality without any fancy processing, just 5 acquisitions, that's it. And this is the fiber bundle analysis. And here is a sacrificed eye, same eye, sacrificed, put it on the microscope, stain it, and then we're able to almost match fiber to fiber. So that's the histological validation, because we have to do. This is the interesting hypothesis that we trying to test when we first published this. So when the tissues were young, the fibers actually align each other because the fibers are not that thick. But the fundus area is limited. When the fibers become thicker, do they stay on top of each other or they push against each other? Which one reflects the truth? Right? At the time, it was like, oh, this is interesting. Can we see it? Now that's the truth. So this is the histology from just one cross-section from here. And this is actually the OCT. So the histology shows that the fibers push against each other, become elongated. And the OCT shows the same, so become elongated. Also, if you look at these fibers carefully, so these are RFL. They're actually 2 layers, right? So there's a bottom layer and there's a top layer. And I asked my friend, okay, do they carry different information? For example, most of them, the bottom layers are on ganglions or off ganglions, or the top layers are on ganglions or top ganglions. The answer I got was no, there will be no functional separation among the top layer and bottom layer, primarily because of the developmental process. The earlier will stay below, the later developed will stay on top. That's the developmental process. So this paper is under review. I think it's under revision. The comments were okay, so we'll be able to publish pretty soon. But tissue is getting a lot more popular. And I'm working with a group of scientists at NIH to develop 2 models, myopic model and glaucoma model. So we'll see how it's gonna work out. And this is another video trying to show. So this is a tissue, and you see that 2 layers, right? It's almost 2 layers. I mean, have you ever imagined the RFL has 2 layers and a different bundle organization? It's actually quite interesting to see. I mean, I've seen this multiple times. So back to the optical property here. We want to measure oxygen consumption. And SO2 stands for saturation of oxygen inside the blood. And that's the beauty for visible light. For a clinical machine, these 2 parallel lines shows that the oxygenated and deoxygenated blood, there's no difference. They're in parallel line. So mathematically, when the 2 features are in parallel, it's unstable to do the inverse calculation. So if you, I mean, in any matrix theory, when you have a matrix, it's non-invertible if the matrix has identity become close to 0. When the two lines are in parallel to each other, means identity are close to 0. But if we move it to the visible range, it's pretty chaotic for scattering and for absorption, which means it has a signature spectrum so that we can analyze to calculate the oxygen content in the blood. This is a major challenging work that we have done. And we have been doing that for several years. I still cannot say we have nailed it, but we made lots of progress. I'll show what that is. So the first thing is numerically, we want to simulate that. We build an eye model. This is a numerical model. And we shine light through the regular path, like any eye, and create a vessel model. So it's a vessel embedded in multiple layers of the cells. And light coming in bounce back at different location and then reconstruct, like I mentioned, that's the OCT process. So this is the simulated OCT image. And we do a so-called quasi-spectroscopy analysis. I'll bypass all the engineering part. So from a mouse eye, take a B scan. So that's a B scan. And then from the B scan, zoom into a vessel. And from that vessel, we do a so-called short time Fourier transform across the whole band, so that we have a OCT reconstruction across different wavelengths. We're not using the whole band. So the downside is we sacrifice the resolution, because resolution is inverse proportional to the bandwidth. The shorter the bandwidth, the worse the resolution. In this case, we sacrifice the resolution, but we gain spatial spectral information. And from the spectral information, we can fit the signature curve. Now the signature curve, you see that? It's the W curve. And if we go back here, this is the inverse W curve. So these two features need to match and then analyze the optical absorption of the blood so that we can quantify the oxygen content. So at the time when Brian Sotigno, he was an MD PhD at the time. He's now a junior faculty at Stanford. This was part of his PhD thesis. Everybody thought, okay, we nailed it. We know how to do it. And then we'll move to human. One of the validation was we have to show that what we quantify are the true values, not the random value, because there's no gold standard. So the gold standard is the blood gas machine. Blood gas machine means we take the blood, run into it's made by Siemens, and quantify the partial pressure of oxygen dissolved in the plasma. And then feeding a partial pressure. So from your physiology course, there's an oxygen association curve, right? Partial pressure is the horizontal axis. The vertical axis is the oxygen concentration. The higher the pressure, the higher the affinity. That's why how the oxygen is combined to the hemoglobin of the red blood cell. We follow that curve, calibrate by the temperature, pH value, anything that we can calibrate. So that's the blood gas machine versus OCT, which means it's quite accurate. Of course, subjected to the precision. The validation is also took us a long time to do. So focus on this curve. The horizontal axis is the partial pressure of the systemic partial pressure. So if you hook up a pulse oximeter, that's what you read as a partial pressure calculated as oxygenation calculated through the partial pressure. The vertical axis is oxygen consumption. Oxygen consumption contains two values: blood flow and oxygen concentration. So from here, we reduce the systemic oxygen content of rat. So the rat consumes less oxygen. We just supply less oxygen to the rat, but the eye consumes more. So let me repeat this. When we provide less oxygen to the ventilation system, the eye consumes more. It's counterintuitive, right? When we provide less, the eye is supposed to consume less. So it puzzles us for a long time. And my colleague in the department, Professor Robert Linsenmeyer, his whole career in the past 30 years has optimized a technology using a silver chloride needles that poke into the eye. So this is a bloody experiment. Poke into the eye, measure the partial pressure at different locations of different cell layers, so that we can build an analytical model to map the partial pressure and estimate the oxygen consumption. So the red curve is the model estimate. The black curve is the OCT, visual OCT measurement. They agree. So something must be not so wrong. So it turned out to be when we supply less oxygen to the animal, the choroidal circulation provide less oxygen because the choroidal circulation is never regulated. It's the same as systemic circulation. When the systemic partial pressure drop, choroidal drop. When the systemic increase, choroidal increase. Now the systemic drop, so choroidal drop. The most demanding area is actually the outer retina, the photoreceptor, the bipolar. It's take the oxygen by oxygen diffusion. When the pressure drop, the cortical circulation provide less. But the eye is highly regulated, similar to the brain. So the irregular circulation needs to compensate for it. So we actually quantify the compensation. So this is actually quite interesting. Another case is oxygen consumption in a diabetic model. So this is called Akita TSP. Akita is a type 1 diabetic mouse. TSP is an anti-angiogenic gene being knocked out. So in this case, this animal has severe angiogenesis at the week of 25. All right. So focus on the timeline. At week of 25, according to the literature reports, the eye will suffer severe angiogenesis, which will have potential hemorrhage, and that's the downstream of the retinopathy. We imaged the group of animal at 5, 7, 9, 11, 13th week. So these are considered healthy at the time. And we validated from 5th week and the 13th week, there's no capillary change. The body weight keep growing because they were young mouse, and the glucose level keep growing. So diabetes is getting severe. Here's the interesting part. The IMRO2 is inner retinal oxygen consumption. For a healthy one, they maintain roughly the same. For diabetic ones, they increase constantly. And around week 11, the differences of the oxygen consumption become statistically significant, that the diabetic eye requires a lot more oxygen. This actually partially explains that why diabetic retinopathy is a ischemia-driven process, that anti-VEGF may treat the symptom, but will never treat the reason, which is the oxygen consumption issue. So like I mentioned, when Brian finished the rodent study, we believe we know it. We want to do human study. We had so many headaches, like the human oxygen consumption measurement was never stable. There were all sorts of headaches. The cataract kills it. The motion kills it. There's so many headaches. How many headaches there are? These are all the headaches. So in one of the Thanksgiving party, I asked my team members to come to my house earlier. The party typically starts at around 5:36. We start drinking and all the way to the midnight. On that particular 2018, I remember that 2018. As I come to my house earlier, I reserve a room in the local library. I ordered Papa John's pizza. I said, okay, so we just lock everybody until 5 p.m. and we have to list all the problems that you can think of that contribute to the instability of the human oxygen concentration measurement. So these are all the things that we list, like the top wall normalization, polarization of the light, motion, back, you name it. So we list so many, and then it was pretty defeating because there's so many things that we don't know. After a few years, the gray means we solve it. The white means we have not started. The yellow means we partially solve it. So I think it's pretty good. Most of the things are nailed out. So this is the latest paper actually just being published. We have developed a new iterative algorithm that the stability of the eye is actually being overcome. So the current work right now is we're trying to apply this, working with colleagues to quantify the oxygen concentration and potentially consumption of glaucoma at different stages. So this is ongoing work. So move to the front of the eye. This is something I mentioned, right? The outflow pathway controls the fluid being pumped out. When the outflow pathway has certain blockage, the fluid cannot be pumped out at the sufficient rate, so there will be build up. The build up increase the pressure. The increased pressure will have all the headaches. So this is the visualization of the pump out process. When the fluid is being generated by a cellular body, it will go through this path and then being pumped out through a trabecular meshwork. Is like a sponge. And through the trabecular meshwork will be collected by a ring type of circular structure. It's called Schnapp's canal. And then from the Schnapp's canal, there will be collector channels, just like different valves. And the collector channels just drain the fluid outside to the episcleral vein, and it's actually being just, when we blink the eye, it's pushed out. And this is the top view. So if this is the pupil, this is the Schnapp's canal, and these are the limbovascular network that connected to the collector channel and drains everything out. So anything can go wrong through the process. People believe that trabecular meshwork contribute the most resistance of the outflow pathway. Of course, people are trying to visualize that. So this is an electron microscope showing the limbo network and showing the Schnapp's canal. And this is my colleague at Northwestern, Ben Thompson. They visualized the similar network using antibody-based staining of confocal microscopy, but none of them are in vivo. I mean, whether in vivo is possible or not, let's see. So because we build OCT, so we actually, in our small animal OCT, we mount a circular motor and just rotate it. And this is the rotated OCT angiogram. And this is the Schnapp's canal. Now you see that, right? So it's a circular structure. At the time, we have to manually stitch them, which is fine. As the proof of principle, all these are acceptable. And when everything is visualized better, you actually show that this is the, so the gray, like the sausage shape, this is the Schnapp's canal. And all these vessels are limbo network that drain everything. So at the time it was 2D. Lisa Beckman was the first author. She did a beautiful job. And one company hired 2 students from my lab last year. Have you heard of a robotic surgery called Da Vinci? It's made by a company called Intuitive Surgery. I'm sure they're gonna do eye surgery because they hired 2 students from my lab in the same year. Both are in the technology department. So also I want to show that the Schnapp's canal is not a smooth tube. It is never a smooth tube. Lots of literature and mathematical models made an assumption that it's a smooth tube. And I argue to the field that it's never a smooth, it's actually an ugly structure, serve a purpose. So in other words, when anyone doing investigation of the Schnapp's canal, you cannot rely on a cross-section at different location because it will change so much, location by location. We have to image either a whole circle or a big portion of it. I'll bypass it. So a nicer way to acquire the image and then also setting the stage for potential human translation is mount everything on a robotic arm. So this is a robot and it's scanning. So on the right side, that's the robotic arms controlling of the robotic arm. And on the left side is the OCT on the fly. And when the position is right, now you see the real-time OCT image. And this is the pupil camera showing the pupil as the eye is being scanned. The OCT is about the size of 2 phones. So just mounted on a robotic arm has to be as light as possible for the accuracy reason. So this project is led by Raymond. Raymond is a MD PhD student, will become an eye doctor someday. just show the results from last week. robots is also never accurate if we rely on robots for surgery. It's accurate enough for car assembly because the requirement is at the level of millimeters to centimeters. For the eye surgery, we need a requirement accuracy of microns or submicrons. It's a different scale, so it's 1,000 times difference. So we have to develop tools to align them correctly to provide any surgical guidance. So Raymond showed me a so-called point cloud registration. So what is that point cloud registration? For the new phone, when we look at the camera, the face lock, the face ID can unlock the phone. What is the face ID? They actually have different laser scanning the face with different cloud being captured. Similar as the autonomous drive of Tesla, they have laser scanning around all the locations and get the cloud and then register. So we're trying to mimic the same thing. Now you see, this is actually the whole eyeball scanned by the OCT across the whole mouse eye. The registered, so on the left side, this is the registered whole anterior segment. And now you see on the right side, these are fly-throughs across different cross sections, either sagittal or coronal. Took us a long time to figure it out. This is another video to show the overall visualization of the whole eyeball. And this is OCT of the limbovascular area. And why we even bother to do all of this. Eventually, we want to map the whole synapse canal. And once we map the synapse canal, we want to visualize the collector channels 1, 2, 3. We want to map the collect channel around the whole 360 so that we know where the concentrations are. Each patient will be different, and this will provide a guidance for the stent surgery. So that's the goal, and that's why we're building a robots. Eventually, we want a patient sit there, robots just scan across. That's it, and we'll provide a objective patient-specific guidance for how many stents needs to be injected and where the stents needs to be injected. So back to the human work. In 2013, we published the first first animal, and then in 2015, the lab published the first human study. But I want to show you how hard it is to build a machine that can be operated by people not from your lab. So the PhD students they're genius. They can do anything with a craft. Now in 2016, we package everything into a big cabinet and moved to Northwestern Hospital. And we let the photographer to use. It was a nightmare. And so the system broke almost weekly. In 2017, so I started a company, and I believe letting the company taking over the engineering process and the lab focus on the science is a better separation of the focus. So this is the first prototype of the company. Still, it broke every week. 2018, the second generation. Now from here to here, sounds like a small change. From here to here, it's a huge change. Look at the big thing, it's packaged into a small box. From here to here, the footprint remain comparable, but the eye fixation, the optical design, the software become a lot better. During COVID, that was the third generation, and we made lots of mistakes. Any mistake possible, we made lots of mistakes. In 2021, that was the latest generation. So this is the latest generation. At least the footprint, the appearance, they're all the same. So we fixed that. And right now is more software, usability, user feedback, this type of work. So again, you see the picture of GE. Now this is the 2015 paper. I want to look at the time. This is the RNFL from a volunteer, and it shows that the RNFL oscillation is actually visualized by the higher contrast and higher spatial resolution. Ian is another one who were hired by Intuitive Surgery last year. He proposed a high definition speckle reduced scan. So in other words, if we oscillate the optical scanning as compared to a single line to acquire a B scan, and we can synthesize these pixels to reduce the speckle. So you will see what does that mean. So this is a regular B scan along the straight line. The quality is okay, is acceptable. But look at the greeny structures. So these are speckles. Speckle is unavoidable in OCT. There are ways to suppress it, but there's no way to get rid of it. And speckle is more prominent in visible OCT because of the enhanced backscattering coefficient. Now, with his method, that's the resolution. So look back and forth. So the speckle noise has been significantly reduced just by oscillating the scan rather than a straight line, just oscillate a little bit. And okay, now let's look at the real meat. Aurora is the visual CT machine. And you probably know many of the names, right? Trident, Circus, AngelView, and Spectralis. These are images acquired from the same volunteer within the same hour in the same room. So we show the quality. And that was the 2020 results, not even the latest one. The latest one is even better than that. So the outer retinal layers, look at the COST and RST. It's called raw and cone tip. All these were only visualized by adaptive optics within a small field of view. But in visibility, you just get it. It's there. And it's up to the physicians to, Chris, it's up to you how to use it. I'm told that the ROST and COST, when the two bands disappeared, is one of the earliest signs of aging. I'll show another case here. The synaptic layer. So at the inner, at the IPL, here is not so visible. But if we process better, in other words, reduce the speckle more, average a little bit more, what we can see is here. The IPL has 5 stripes. These 5 stripes corresponding to the ganglion and bipolar synaptic connections. They form at different layers. And people believe that the off-ganglion stays below with respect to the light path, and the on-ganglion stays on top, and in the middle is more or less on and off ganglion. So now we can visualize this to correlate the on and off ganglion at different stages of glaucoma. The ganglion cells are not dead. They're still alive, but the synapse retracted. So they become non-functional, but they're not dead. So the RFL may not change. The fiber is still there, but the synaptic connections are gone. So they're not responding to the light stimulation. So it provides a way to investigate the glaucoma at potentially the earliest possible stage. Another headache, like I mentioned, getting a system from scratch is really challenging. I call Zeiss, I call Heidelberg, I call all the major manufacturers, hey, can we collaborate? They were no, we don't care. And I said, okay, that's fine. We'll do it by ourselves. So the speckle were down, but the human scan needs a higher speed. At the time, so the image I showed you, these were acquired at 40 kilohertz. Commercial OCT start from 100 kilohertz up to probably 400 or 500 kilohertz. It cannot go too much higher because of laser safety. Now, if we just do 125 kilohertz, this is the quality. What are those things? This actually contributed by a noise called a relative intensity noise. This is intrinsic to the laser that we use, but unfortunately, that's the only laser possible that we can use for visual CT. So we'll have to find a way to get rid of that. Again, Ian made a great contribution. to design a new OCT. So of course we have to patent that. So the whole OCT structure is is not a microcell anymore. It's a microcell plus a Mach Sanders and using two spectrometers to cancel each other. And this, there's lots of math behind it, but it's very effective. Again, this is 125 with one spectrometer. This is the same 125 with two spectrometers. I mean, again, the quality just day and night, right? So speckle, motion, speed, we just solve them one by one. There's just so much work. And it's actually set the stage for clinical study using the OCT, because we deal with the engineering part. This is another image. So this is the fundus image. And with the balance, you just show the quality is just much better. Again, this is the final machine. Quite a few places start to work with us to test it. And there will be one here, not necessarily in this building, but at HKU working with Chris. So let's move to super resolution quickly, and then I'll show why we like it and where's our contribution. Super resolution is a 2014 Nobel Prize. Eric Benzik from Howard Hughes and Stefan Hill from Max Planck, and there's a third one from Stanford. They shared Nobel Prize, but behind the scene, Xiaowei Zhang from Harvard and Howard Hess from Howard Hughes, they also contribute significantly to super resolution microscopy. They were not awarded Nobel Prize. Nobel Prize can be awarded to up to 3 people. So that's too bad. What is that? What is the localization microscopy? The resolution limit is called diffraction limit. Diffraction limit means the ultimate resolution is controlled by the wavelength and numerical aperture, but more of the wavelengths than numerical aperture. So you cannot resolve a object less than half of the wavelengths. That's just a physical limit. How to overcome that? So here's the principle of the localization microscopy. There are 5 molecules that are so close to each other. So optical microscopy can potentially tell there are molecules there, but how many and what they are, that's impossible. If there is a photochemical manipulation, that these 5 molecules won't emit fluorescence at the same time. Only one emit at a time. Let's say this one emit at a time. So if we know statistically there's only one molecule emit the light, we can bypass the size of the spot. We can locate the center. And we repeat the process. So there will be multiple localizations. That's why it's called localization microscopy. The downside, so that's Nobel Prize. The downside is how to tell whether they are same species or different species. So my lab contributed to optical design that we introduce a component, dispersive component, either a prism. So prism is dispersive component is, if you put prism under the sunlight, you will see a rainbow. Why that's the case? Different wavelengths will go different paths in the glass because of the reflection index is wavelength dependent. Same thing here. So if we introduce a dispersion component, we'll have a rainbow. If it's the same color, they just go different paths. So we'll have a localization and we'll have a optical signature associated with it. And we do a numerical combination. This is the on the fly. So on the left side, this is the random signal. Each blinking corresponding to a single molecule generated fluorescence light. And on the right side, these are the spectrums per molecule. And based on a mathematical model, we can reconstruct the molecular information from where they are, and we know what they are. This one shows that if we don't have the spectral information, people may believe this is a single molecule. But based on spectrums, there are actually 2, just very close to each other. And this is a cell image. We lost the competition, so we were sending to Nature, and we were rejected, and we sent it to Nature Method. We were rejected again, not because the quality. I mean, Nature was the quality. Nature recommended us to send it to Nature Method, but Nature Method said somebody else publish it. We start a Berkeley group published that. We started around the same time, but we never learned how to do cell staining because it's everybody in my lab are either physicists or electrical engineers. So I have to work with Howard Hughes colleagues to do that. But at the end, we publish it. What is that? So these are molecules that the fluorescence emissions are almost the same. So using traditional optical method, they are the same. You cannot separate them. But because we know the whole rainbow, numerically, we know they're different, and we can reprocess that. So that's actually the beauty of it. And what's the indication? The number of different labels are unlimited because we just numerically analyze all this. Without going into the details, how are we using this? One study is quantify the mitochondrial damage in cancer cells in diabetic retinal cell, epithelial cells. Another study is chromatin imaging. So these are whole nucleus that we quantify the histone markers that are associated with epigenomic stimulation that when we apply chemotherapy, for example, trying to understand why different cells survive the chemotherapy, why different cells can be killed by the chemotherapy. What you see here, this is the primary culture retinal ganglion cell from a mouse provided by a colleague from Stanford. What we're trying to do in this particular case is to visualize the chromatin structures in the soma to predict how likely the cell can survive after implantation. This is something interests lots of colleagues in chemistry field. I want to give you a hint and then say whether we can, going forward, we can work on this potentially. So Alexa Fluor 568 is a molecular dye people use widely for superluminous microscopy. And we label the cytoskeleton of inside the cell. So it's the same dye. It's the same cell organelle in the same cell, but the color looks different, right? It's the same dye, same cell, same cytoskeleton, but the color looks different. Why? What makes them different? Essentially, what we get is if any of you are doing the fluorescence microscopy, you will get an excitation spectrum and emission spectrum from the manufacturer. But if we look at the single molecule signatures, they're actually very different at the single molecule level. Just like if an alien look at us, we're all human beings. But when we look at each other, we're also very different. I think that's the same thing. Each molecule is actually different, even if the same molecular species. So we're trying to match the numerical simulation with the studies, and we match it. And thanks God, it's very interesting. So Zhang, he was leading this study. He now started his own career as a junior faculty at North Carolina State University. One last example. So multi-scale, all right? I talk about OCT, I talk about super resolution. What do I mean by multi-scale? So this is a corneal damage study. What we did is we take control mouse and we take mouse that we artificially increase the intraocular pressure. So it's a hypertension model, but it's an acute hypertension model. There are two groups of mouse. One group is treated with ROCK inhibitor, ROCK kinase inhibitor, which is very popular in glaucoma and trying to reduce the pressure or maintain the pressure. But how the pressure is maintained and what's the downside of ROCK kinase inhibitor to the cornea, because it's topical application. So on the left side are OCT images. So this is a healthy cornea of a mouse acquired by OCT. And this is the OCT image of a cornea that's heavily edema, where we increase the pressure without applying the ROCK inhibitor. This is ROCK inhibitor with normal pressure. This is ROCK inhibitor with high pressure. Now you can see edema is a lot less. But why? What contribute to the reduced edema? The work that we did here is we define the tight junction to 4 different types of the endothelial cells. Their type 1 means healthy tight junction. Type 2 is distorted but still maintain the tight junction. Type 3 is a cell has partially distorted, so the tight junction is destroyed at certain location, but not totally. And the type 4 is just tight junction is being pulled apart. And we validate using super resolution, and we validate using electron microscopy. And look at that. Because of the confocal microscopy is large field of view and defined by the super resolution microscopy. So we can do a statistical analysis. C1 means control normal pressure. C2 means control high pressure. C3 means rock normal pressure. C4 means rock high pressure. And these are 4 type of tight junctions. The green, the type 4, which is the worst one, doesn't exist in normal one, doesn't exist in control in the rock normal pressure, which is expected. But look at the percentile of type 4 tight junction in the control high pressure. So much more, right? And compared to the rock inhibitor high pressure. So the rock inhibitor maintain the tight junction and reduce the population of the fully distorted tight junction at a much, much lower, which is almost 5-fold reduction of the type 4 tight junction. It's actually partially explain why the rock inhibitor is so popular. And actually certain protection mechanism behind it. Okay, I'll stop here. I talk too much. We build OCT, we build super resolution, and we combine them to investigate multiple diseases. We focus on small animal, tissue, and heavily pushing clinical study in the next several years. This is the campus. My lab is somewhere in the basement over there. And this is my jogging trail along the small lake. So the one full circle is 1.1 miles. I typically run 2 miles, 2 circles. And this is our student gym. So I go shower and then go to work, which is perfect. Thank you.
So we truly thank Professor Zhang. The first paper on OCT was published in, anyone else? 1991. So more than 30 years ago. And you can see Professor Zhang has perfected the instrument in many ways. I met him last year during a conference in Jerusalem. We chatted, we talked about OCT, and I just casually asked him, well, I mean, obviously there are many groups now in Europe, in the US, working on OCT, building instruments like what he's doing. And I asked him, which group overall you think is the best in OCT? This is what he said. We are the best. And so it's interesting to see and fascinating to see how engineering can work hand in hand with ophthalmology in such a beautiful way that we can now using better resolution, high speed, and high and tools to allow us to study biology of the eye, to study diagnostic aspects in many different eye diseases in ways that we wouldn't be able to do before. So just a few questions regarding the talks you deliver. Now, I have to admit that there are obviously gaps, or there are space that you're working on, you're building this up. And in terms of the very beginning, you mentioned about the two layers of the retinal fiber layer. Do you see similar structure, 2 layers, humanized? And you mentioned briefly that it doesn't appear to be a functional correlation. We don't know yet what it means, but would you like to speculate what would be like in human eyes, and how would the technology give us a better understanding of the retinal nerve fiber layer?
So I was told, again, I don't have credit in this area. So I was told that the bottom layer is closer to the optic disc. Those fibers are closer, and then the earlier development. And the top layer is actually far away. And because, I mean, the closer they occupy the space, and the further periphery ganglions, they have to combine from the top area. I don't know why it's on top, why not squeeze at the bottom, but that's what the speculation. I asked so Yang Hu, do you know him? He's from Stanford. He's one of the leading RGC biologists. He said there's probably no functional differentiation between the two layers. He doesn't believe that, but it's geography related. That's, yeah.
Right. So from the work that we did here, retinal nerve fiber optical texture analysis, which allow us to see the bundles structure of the retinal nerve fiber layer, we see in human eyes, the retinal nerve fiber layer can be visualized in different bundles, different layer of bundles. And now we don't have a human eye here, but the animal eyes we see, do you think it's not two layers, but in fact it's different types or different architecture of the collection of the bundle? So you have, I see there's like you have collection of fiber denser of the upper layer on certain location, and then the next pile would be a collection of bundles which is denser of the inferior layer. So you think it's an architecture arrangement rather than having two layers of bundles?
We didn't see that in mouse. There's a small study here. So what is that study? This study is a nerve crash model, right? So you clamp the optical fiber bond, the nerve bundle behind the optic disc, and after a few days, half of the RGC will start to die, or a certain portion of the RGC start to die, and we follow that portion of the RGC from day 0 to day 15, and they essentially die out, means the RFL thickness will disappear. So if there is a different type difference of the fiber bundle, they may have a different dying rate. We didn't see it. We actually see the fibers become swollen in the beginning. So it shows here, become swollen. So what is this curve? This is the statistics of the mean fiber diameter. The mean fiber diameter become bigger and then become smaller. So why that's the case, I don't know, but it's being observed, and it follows quite a few hypotheses in glaucoma as well. In the earliest glaucoma, the fiber diameter is actually increased, according to what I've read. But there's no type specific change, or at least we don't know that yet.
In human eyes, we see different types of bundle. For example, the papillomacular bundles, the bundles serving the macula, they are typically smaller in diameter, and is different from the, say, arcuate bundles, which the O4 fiber diameters or bundle diameters are bigger. So there are intrinsic variations in the arrangement, in the trajectories, in the diameters, in human eyes, and obviously the mouse eye, you have a very thin retinal nerve fiber layer. That's why you don't see these two layers. In human eyes, in tissues, in non-human primate, the retinal nerve fiber layer is thicker, and that can results of the bundles, obviously depending on where you image these bundles. The closer to the optic disc you are, the thicker the layer, and then you get a different stack and architectures, organization of these bundles around that nerve head.
I think I get your point. I think for the tissue one is if we create a model, we don't have a model yet. We don't have a glaucoma model of the tissue yet. We might have a better sense of how they distribute in the glaucoma area. Yeah, I see your point. So all this I show, these are healthy, happy, they may be happy, they may not be happy, but healthy, normal tissues.
But that definitely is a beautiful image, and thank you for sharing that. Now, another question about, I think it's also interesting to see how engineers can use technology to ask questions that we all are interested to know as ophthalmologists. You mentioned the pathogenesis here about diabetic retinopathy show is the increased oxygen consumption. Now, from your work, can you speculate or can you tell us where is the oxygen consumption in the retina? Where, for example, which layer of the retina or which type of cells that cause an increase oxygen consumption?
Okay, pure speculation, no scientific evidence, right? Mitochondrial damage is created by, can be introduced by higher glucose. So in this case, the mitochondrial damage cannot perform anaerobic conversion to provide enough ATP. That's universal. Because of that, they have to do aerobic metabolics to provide sufficient amount of ATP. That's where the demand is up. If it's cell type specific, which cell type consumes the most? The photoreceptors. So most likely through the disc shredding process, right? That's actually at the night being regenerated. That's where eye becomes most metabolically active. And that's when the eye requires the most oxygen. And at the time of the glucose insult, the vascular system cannot provide sufficient amount of oxygen. Right. Pure speculation, no evidence.
So Amy is our vision scientist, and she has a lot of ketomites. Angie is our VR specialist, and every day we perform laser surgery for patients with diabetic retinopathy to destroy the photoreceptors in order to decrease the oxygen drive.
Right.
So perhaps, do you have any idea after listening to Professor Zhang's talk? So he showed the increased oxygen consumption in the retina in diabetic
early diabetics.
So Amy, you have any thoughts on that?
From what I know, when there's hyperglycemia, when there's increased glucose, actually there is another pathway to generate ATP, which is the aldose reductase pathway. Yeah. So through the aldose reductase, then glucose can be consumed to generate ATP. So I'm not sure if that's related. Yeah. So do you really need to consume more oxygen when there's hyperglycemia? Because
the second pathway will compensate for the ATP production. That's
like, yeah, shunting the pathway towards this second, say for example, way to produce more ATP. So that's what I'm thinking.
I see. Again, it's possible. I would say, I don't think, or at least based on my reading of the literature, it's clear that whether there's a type specific increased demand. But the majority of the oxygen or the ATP demand is actually from photoreceptors. And and most likely it contributes to the largest percentile of oxygen consumption. So you always start from the highest consumers.
The idea here is we know injecting retinal regulation, the ultimate goal of this therapy is to decrease oxygen drive. I think we think less often about why there's increase in oxygen demand. Okay. So Angie, any thought on that?
Yeah, echo your thought. Actually, you made a well. First of all, congratulations, Professor Zhang, for your many wonderful advances and your OCT is like I just got blown away by the high resolution. I mean, I'm using Spectralis daily, and I thought it was a pretty good machine until I saw your images. So I'm looking forward to trying Professor Zhang's OCT when it arrives in Hong Kong U. But I echo Professor Leung's comment. You made a comment just now about the treatment, about the oxygen consumption, and you say that anti-VEGF actually only treats the symptom, but not the root cause of the diabetic retinopathy, because it doesn't address the problem of the high oxygen demand. And in clinical practice, and that's I echo that because there are clinical trials using anti-VEGF as the primary treatment for diabetic retinopathy, and I've always have some reservations as to whether it actually works. But for us, the mainstay is still PRP, but PRP actually works by reducing oxygen demand, by reducing it. So I feel that that's why in my clinical experience, PRP actually works. But anti-VEGF, I mean, yes, you have to inject it many times, but I always still have this reservation of whether you can actually treat the root cause, and your comments actually kind of gave me more insight into that. Yeah, so
I think the philosophy, I mean, totally speculating. Philosophy will be as long as the eye won't, as long as there's no hemorrhage, the eye should be okay, regardless of whether there will be angiogenesis or not, because it's the hemorrhage kills the vision. So just prevent the hemorrhage. And as long as possible, somebody else will kill the patient.
One final question for me. Now, you show beautifully that the super resolution microscopy is useful to delineate better the structures of the tight junction. Can you go back to that slide? I have a question on that. Now, based on the conventional fluorescent microscopy, we can already see some differences. The 4 types, top panel. I'm pretty ignorant on the second panel here. What are the structures and how much information the super resolution microscopy add on top of what we can see already with conventional microscopy?
Yeah. So I get your question. So question is, what additional information super resolution provided as compared to confocal?
In this particular panel.
Yeah. So in this particular case, the super resolution provides a quantification of the level of damages at the nanoscopic level. And that's the major value, especially because the damage is a continuous change. There's no gap. So there are 4 types, but how to define the 4 types that made sense? We have to do a statistical analysis. And the statistical analysis requires the highest possible resolution to define the distance being.
Right. So correct me if I'm wrong. So we're looking at the gap of the tight junction between 2 cells.
Yes.
So this is what you're measuring.
Right.
Right.
And this is more of a research tool. And if we validate that's the case, the super resolution is not necessary. So that's one case.
Provides insight to the biology of the process ongoing.
Right. And I don't think super resolution will have any clinical value. It's a multi-million-dollar machine, and it requires very sophisticated sample preparation. It's only investigated at a single molecule level, maybe single nucleus half of cell, it will have very big challenge to image even a small piece of tissue slice. So they serve totally different purpose. So.
Right. So I think we all have a very good understanding about what the work of Professor Zhang, and we're grateful for his lecture.